Visionaries is proud to present its 24th season on public television. One of the great wonders of science is the window it has opened into the microscopic world. There, inside all living things, an epic struggle is always underway. Bacteria invade the land, viruses scale the gates, and cancers claim territory. It's epic. And as such, there are valiant, heroic defenders of the realm of the living, our immune systems. Here's where the story gets interesting. Our heroes have a secret weapon. It's called science. Imagine if we could decode the immune system to unlock new preventions and treatments for some of the world's most devastating diseases. Imagine a life expectancy of 125 years or more, with no sick days, not even one doctor's visit or days spent in a hospital. This is La Jolla Institute for Immunology's mission in a nutshell. Every day, its researchers work towards making discoveries of new weapons in the epic struggle against disease that will lead to better defenses and ultimately cures for immune system-related diseases.
The immune system touches almost every disease. It's hard to find a disease or a condition that's not affected by the immune system. And why is that? The reason is that your immune cells are everywhere in the body. Some of them are circulating and patrolling everywhere, and others are actually resident in your kidney and your intestine. There are immune cells that reside there and contribute to the overall tone of the tissue or function of the tissue, as well as fighting infection. The work here, we divide into three categories. There's infectious disease, which is very important, and vaccines, which are the most effective public health measures. But we also work on cancer, and stimulating the immune response to fight cancer has become a burgeoning area of new therapies that are less toxic and in many cases more effective. And also equally important is learning how to turn down those inappropriate immune responses, particularly in the more than 80 autoimmune diseases, some of which attack particular tissues, type 1 diabetes, for example, the pancreas. Our mission is to make fundamental, great discoveries on how the immune system works to the benefit of human health. And therefore, we've adopted a motto, life without disease, which is a direction. When you first hear it, it makes you think, which is good, because presumably there will always be some diseases, but we can have life without polio or life without smallpox. We've reached that already. Why not have life without HIV, AIDS, or life without malaria, or life without tuberculosis?
This structure is from X-ray crystallography. Immunology is a beautiful and complex science. It's the science of understanding how your body can heal itself, how every day it detects and destroys invaders before you ever know you've been infected, and how it detects and destroys cancer cells before they ever take hold.
And this one's the light chain, and that one's the heavy chain. Is there more contact of the light chain and the heavy chain?
And you can study immunology and when it goes right and when it goes wrong on multiple different levels. You can study it at a population level, how we develop herd immunity and how we develop or don't develop diseases. You can study it at a whole human level, how this one human has conquered an autoimmune disease or how this one human has fought off an infection and become immune. You can study it at the cellular level. I study it at the far extreme, the molecular level. Viruses are, at an intellectual level, fascinating because they can be so simple. So you and I have 20,000 genes in our human genome, and that makes 20,000 molecules for 20,000 functions. But these viruses are incredibly simple. Ebola virus has just 7 genes, the fever, 4. Exactly 4. So it makes 4 different molecules. It's a machine with 4 working parts. And so one question is, how can something so simple be so pathogenic and so deadly, and how come our much more complex selves and our human intellect are powerless against this tiny, simple thing? But on the other hand, if the machine is that simple, we can take it apart and we can examine it. And there are vulnerabilities there, and that's exactly what we've been able to do. What we're using here is not microscope, but microscope. The one we use is 11 feet tall, and it has a massive and incredibly powerful beam of electrons operating at 300 kilovolts, focusing incredible illumination on a very small biological sample. And it has sophisticated electronics that allow all of those electron beams to be aligned and refined to get the perfect image. We've been able to understand how these very few molecules are folded up and how they work. We can see where they change and where they can't, and we can see exactly the shape that no one has ever seen before. We had developed a body of work where we'd finally cracked the structures of the key molecules of Ebola virus, Marburg virus, Lassa virus. And the field now, for the first time, has life-saving therapeutics against Ebola virus. It's not hopeless. The most powerful microscopes commercially available are focused here at Jolla Institute on coronavirus to understand the molecular nature of the atomic bonding by which that virus is built. If we can see it at that level, that is the complete roadmap that we need to understand how to defeat it. The Coronavirus Immunotherapeutic Consortium is designed to unite all of the antibody discovery labs around the world to figure out which of the antibodies we are all making are the most potent against the virus, which ones should be delivered as a drug. And this is important because even once there is a vaccine, it's not going to be in all places at all times. There will be people that haven't yet been vaccinated. There are people that can't be vaccinated. We need to protect people while we're waiting for that vaccine. If we can find antibodies, we can give them as immediate and safe drugs for protection. But we got to find which ones are best.
So this is our beacon instrument, which allows us to look at immune cells that make molecules called antibodies, which precisely target particular sites on our pathogens of interest. So what this instrument does is identify where individual immune cells are located on this little chip, puts a light box around it, and moves that cell into these holding pens. Now you can see it shifting this one single cell now out of the pen, and it'll put it into this little channel and then export that cell into a plate where we can grow it or isolate the DNA that has created that antibody.
Our goal is to make sure that no one is priced out of these therapies, to look among the ones that are being discovered, to figure out which are the most potent, which ones are the most scalable, to make sure that we can deliver immune protection broadly around the world. Because this virus anywhere ultimately becomes this virus everywhere.
I'm obsessed with vaccine immunology. Vaccines are extraordinary medicines. Vaccines are probably the only medicine that's recommended for every healthy person on the planet. right? That's something special to be able to say. And they've just saved really extraordinary numbers of lives. And in general, it's not even noticed, right? Because you give vaccines to a healthy person and it keeps them from getting sick, so you never see the sickness at all. The measles vaccine alone has saved 14 million lives in the past 10 years. That's an incredible number, and that's just one of the 26 or 28 vaccines. It would be fantastic if we could have more vaccines that could do those types of things for improving human health. It's easy to think that, well, we've made at least a couple dozen vaccines against a variety of things, so why isn't it easy to just keep making vaccines to other things? Many of those vaccines were developed before we understood much science, and certainly before we understood the immune system. I would say most of our understanding of the immune system has come since 1950. Most vaccines were actually developed before 1950. And so vaccine development grew as a very empirical trial and error field. The classic way to try a vaccine is somebody has an idea about what particle, what thing they want to try as a vaccine. And essentially, you try it and either works or it fails. It's an all or nothing, okay? And if it works, great, that's a huge success. But if it fails, you've really learned nothing. It's just empirical, there's nothing to build on. And so we really want it to be much more of an engineering problem, right? Where you can try something and you can fail, but you can learn from that failure. Over the past 30 years, we've learned an extraordinary amount about the immune system, all the way from genes and proteins involved to the many, many different cell types involved. And so my big focus, how can we really connect all this knowledge we have about immunology with vaccine development? One thing that a whole bunch of labs are trying to do right now with coronavirus is say, well, okay, probably people who are getting sick but recovering are making really good antibody responses. And so if we know what those antibodies are binding on coronavirus and what they're doing to coronavirus, that should accelerate vaccine development, because that gives you something to focus on. What's the right target? The more we know about that, the more likely it is that somebody can make a vaccine that can mimic that. You want to take someone who was infected with coronavirus but got better and take B cells from that person and find the right B cell that's actually able to kill coronavirus with antibody. We want to understand what are the underlying rules or principles of the immune system that control a good response to a vaccine. The guiding principle of the lab is really, we go after whatever we think the toughest problem is, or wherever the biggest bottleneck is in the understanding, and try and answer that so that the whole field can move forward for studying, again, many different vaccines.
I study dengue and Zika viruses, and the number one question we're trying to address is how does the immune system try to contain these virus infections and not cause disease? Because the immune system is involved in not only protecting against this virus infection, but under certain circumstances, it can actually contribute for the people coming down with severe manifestations of dengue infection or Zika infection.
This is Dakar.
And this is Dakar.
I was born and raised in Nepal. Then I came to the United States when I was 12 so that I could get all my higher education done in a Western country.
This machine you can see how many.
I consider myself Nepali American.
how many is the virus to really infect in the cells.
I wanted to do something that would help globally, not just the US and the Western world, but also that part of the world.
What is the real infection for Zika virus in BSK cells?
So when I entered my postdoctoral phase, I decided instead of focusing just on immunology, I wanted to branch out and venture into diseases that really affected that part of the world. Nepal has a huge burden of these diseases that really impacts the people in multiple ways, economic, well-being. So that's when I decided to do a postdoctoral fellowship on virology, starting with the dengue viruses. And that's how I entered the field of this, what we call the flaviviruses, which includes dengue viruses, Zika viruses, West Nile viruses, Japanese encephalitis, yellow fever. So did you have a dengue control too, or just?
Yeah, we have dengue control too, LPS control.
Traditionally in our field, the focus has been on antibodies. What our work is showing is that in addition to antibodies, T cells are really important. On top of that, our work is also showing that antibodies under certain conditions can also play a bad role. And in case the antibodies are not good enough to protect, or in certain circumstances are actually bad and cause the severe manifestations, then the T cells are there to protect. And based on our work, we're saying, okay, let's develop a vaccine that induces both antibody responses and T cell responses.
Oh, wow. It worked?
Yes! So, less activation. There are so few labs who do T cell studies, and now we're moving from mouse T cell response to she's now the first one in my lab to look at the human T cell responses. But she's telling me this is the first time where our hypothesis was that based on some other studies, these Zika infected cells are not very good at stimulating, activating T cells, and that's what she's seeing. And this has really tremendous implications for how diseases occur in these humans and how we should go about in terms of vaccine development. Since the emergence of Zika in 2016, we're one of the first labs to show that the immune responses to these viruses are highly cross-reactive. What that means is that both antibody and T cell responses to dengue can recognize Zika and vice versa. So now what we're trying to do is like, okay, based on the immunology that we're learning about these cross-reactive immune responses, it no longer makes sense to develop a vaccine just against dengue, just against Zika. Because if you develop a vaccine against one of these viruses, that vaccine-induced immune response against one virus, yeah, it could protect, or it could actually enhance the severity of infection with this related viruses. So this dengue, Zika, West Nile viruses, these are highly closely related.
Interferon gamma, IL-2, and also the IL-10 for 3-day post.
So one thing we're super excited about doing in the lab is in terms of tackling this global problem with both dengue and Zika, we want to be developing what we're calling a universal dengue Zika or a pan-flavivirus vaccine. Criticism of, Dengue is considered the top 10 health problems in the world. It impacts almost half the world population, more than 100 countries. The incidence of dengue infection, dengue diseases, it has gone up 30-fold in the past 50 years. And that's because these viruses, dengue and Zika, they're transmitted by mosquitoes. And because of massive globalization, urbanization, climate change, these mosquitoes are now moving into areas where they normally were not before. In countries like Nepal, where you would never think a Himalayan country should have dengue, but now we do have major problems with dengue because these mosquitoes have moved from subtropical, tropical regions of the world into these temperate zones.
To okay, can you generate these figures that we need for the paper? So hopefully when
this is the humanitarian aspect of my research. It's not just pure science, which is, yes, we need to be doing so sophisticated scientific research in labs right here in the US, in Jolla Institute. But my approach is we need to be doing this kind of research in countries like Nepal, like India, and use the local knowledge, local capacity to strengthen this global effort so that scientists in countries like Nepal know how to contain this virus infection.
Australia?
Yes. So that we don't have to deal with these new emerging infectious diseases coming out of these countries into Western world, in North America, and right here in San Diego. So my take is we want to be developing research infrastructure in countries like Nepal.
Resources is very limited in Nepal, so the resources is the one of the most challenging part, and then the skills that meet the standard, international standard of the research work is one of the challenges that should be met by the department. We have 240 samples for mice collected, not for this.
I was very excited when Krishna contacted me because being one of the biggest universities in the entire nation, that means we're working with all these young students. So, and this is what we need to be collaborating with these universities, with professors, students, so that we're changing the whole generation and training them.
As easy as you think to grow these viruses, so don't be frustrated when
it should be equal partnership, and we need local knowledge. Here, I'm here in this country, in San Diego. If there's an epidemic of dengue in Kathmandu, ward number 52, but not in 53, the ecological niche, the dynamics, the mosquitoes, the human cultural practices, I may not be knowing what's going on. That could explain how these mosquitoes are transmitting these viruses.
When you get two of them, I would
Science benefits if you have diverse opinions, people from different ethnic groups, different backgrounds joining in and tackling the same problem. And that actually helps the science move forward.
We are doing the last step of the ELISA. We want to reveal the presence of antibody.
I'm calling for a global effort that is more equitable and fair than what's the current standard now.
You could say that it's an exercise in delayed gratification to be a scientist. On the other hand, if we think in terms of not the next 6 months, my iPhone, in 3 years I throw it away, well, whatever I do with it, and I want another one. But if you think of an effective new treatment for a disease, that can be generations and lifetimes. The impact of the polio vaccine, how do you compare that to, 5G? So, I think we have to take a little bit longer view than what's going to happen in 2 or 3 years.
I do think we can achieve life without many of these diseases. And by putting the right people and the right skill sets and the right tools to focus on those problems, we could achieve life without Ebola, life without malaria, life without Lassa, life without dengue. We can, by visualizing what the virus is and where it's vulnerable and what an effective immune response might be, discover and deliver those vaccines and therapeutics for broad protection. And we can do it more rapidly than we ever could before.
9 out of 10 of our experiments will fail. Like we're going to try a bunch of ideas, and If we're failing 90% of the time, we still might be doing fine. You know, if that 10% shows us the right step for the next direction, that's kind of where we operate. So yeah, you gotta be passionate about it because you're failing 90% of the time, right? You gotta have something that's gonna get you out of bed in the morning, and that is, yeah, vaccines are the most effective medicine on the planet. Anything that we could do to possibly make a new one, you know, is absolutely worth it.
Life without disease. What would that look like for you? At La Jolla Institute, researchers are working toward making that a reality for all of us. For the Visionaries, I am Sam Waterston.
Visionaries is brought to you through the generosity of William Reinsel Trust, George and Judy Marcus Family Foundation, Liquid Net, the Carson Family Charitable Trust, Sanofi Pasteur, the Wallace Foundation, the City Fund, Walton Family Foundation, R.J. Hedges and Associates, Berkeley Lights, Blue Cross and Blue Shield of Minnesota Foundation, Cabot Keller Oak Realty Trust, Rodman W. Moorhead III, Blessing Stern of 1080, the John Lazarich Foundation, John Paul Mitchell Systems, Moxie Foundation, Kenton Alexander Weed, Jay Gerwin Foundation, the Linfest Legacy Fund, and Thermo Fisher Scientific, as well as Orange County Community Foundation, Pfizer, Fernandez Pave the Way Foundation, the Dolphinger McMahon Foundation, American Reading Company, Ballard Spar, BD Medical Pharmaceutical Systems, Diana and Khaled Mirza, F.R. Bigelow Foundation, Fakery Drinker Biddle Enrique, Francis Moran Abbott, Gerard Foundation, the Honickman Foundation, St. Paul and Minnesota Foundation, and Susan and Thomas Dunn, with additional support by the following.